Hi, I'm Jenny Brown with InVitro Technologies, and I'd just like to really briefly introduce you to the Logos Luna 2 cell counter. So as you can see, the Luna is quite a nice little unit. It's got some pretty advanced software though, even for its small size. We have some cell counting slides that I can show you. Unfortunately, I don't have any samples that I can run through the instrument today, but this is the slide itself. This is the disposable slide. You can also obtain a reusable slide from Logos, which is very useful. So the slide takes 10 microliters of your sample, which is mixed with your stains such as trypan blue. This then just simply slots into the space on the front of the unit and it's ready to go. So we obviously have the count function here. The system is set up with a liquid lens for autofocus, so you don't need to worry about focusing it. Once you do hit the count button up here, then it will go through and count the cells on that sample that you have at the time. Obviously there's no cells in that sample right now, so it'll come back with quite a blank image, but that's okay for this purpose. So we can escape out of there. So we can go back out of there, and if you're wanting to review the information that you've just counted, you can hit the review button. It brings up the information such as the total number of cells, the viability of those cells, and the parameters at which you counted as well. So the parameters can be adjusted and it needs to be optimized for your cell type. We do have some recommendations available for the most common cell types, but unfortunately not for everything. But you can adjust those parameters quite easily through the protocol settings here. You can save those protocols, so if you are using one cell type consistently in your laboratory, then you can have that saved for future use so that you know you're getting consistent cell counting regardless of whether you're using it yourself or other members of your team in your laboratory are using the same cell counter. You should all get consistent results. So the parameters that you can adjust are based on the cell type and size, so the granularity, the size. You can gate the counting, so you're not including little bits of rubbish and cell parts that are included in that cell sample. So I hope that's been helpful. If you would like a demonstration of this instrument, feel free to contact any of our life science team. Our contact information is on the InVitro Life Science website. Thank you.